Good morning students, and once again I welcome you all to Misostudy. So in today's lecture, in today's session, we will be discussing about some questions which are important for your NEET exam, and basically we will be focusing on the chapter Biotechnology Principles and Processes. Let me give you one brief introduction regarding this chapter, Biotechnology and Principal Processes. In this whole chapter, we have discussed about the different process by which the recombinant cell is produced. We know that how this recombinant insulin is produced. Different processes are there, you can say the biotechnological tools are there with which what we want a desired character, and that desired character can be introduced into any of the cell and we can prepare a copy of that. How we can do that? We can take out a gene, and we can take out the gene with the help of the various enzymes we have. Then what we will do? We treat that gene, then we take another vector and we treat that vector as well as that gene with the help of one restriction enzyme. Same restriction enzyme can be used for both the gene, both the DNA I would rather say, because they will produce a same kind of sticky ends. When they produce the same kind of sticky ends, what I will have is a recombinant plasmid, and that recombinant plasmid is what? It is a vector, and this is used. That is used for basically what I will do, I will take the recombinant vaccine, I will allow that it should enter into our cell, that plasmid free cells. Later on the various copies of that particular cell will be produced, and the desired product, for example insulin, any hormones like growth hormone, they can be produced in the bulk amount. Similarly, various another importance is also there, as per we can provide a resistance in case of the plant. Insect resistance, some pesticides, some insect resistant plant also we have already created, and for example we have created the BT cotton, BT brinjal, etc. So all of these they come under the biotechnological principle and processes. I have taken around 10 questions, and in that 10 questions I have taken the whole important processes which is important for your NEET exam. We have talked about the restriction endonuclease, the type of restriction endonuclease, why they need a specific site to cut, what is a prerequisite for that DNA fragment, that it should be like this, there should be palindromic sequences. I have taken the questions on that. Similarly, we have I would also like to discuss some questions which are related to the polymerase chain reaction. So we will start one by one. Let's start the questions. Okay, this is the first question. In this first question, it says that the figure below is a diagrammatic representation of E. coli vector PBR322. Which one of the following options correctly identifies the certain components? This is a question which was asked in your previous NEET exam, and they are related. They are saying that they have used one vector. Now what is a vector? Vector is nothing, it's just like a vehicle that help in transferring a gene from one place to another, right? Similarly, if in a technical language if I say, that means it is a vehicle that help in carrying that foreign gene of interest. So they have taken one E. coli vector, and the name of that vector is PBR322. Which one of the following options correctly identify the certain option? When I was discussing the whole chapter, I told you that in a plasmid the different sites are there. Some sites are origin of replication, some sites are having proteins for where the replication starts, some sites are the site of these restriction enzymes are there. And what do you mean by this AMPR and TETR? That is a resistance for the ampicillin and tetracycline, right? And this act as a marker. What are these? These are marker, these are restrictions enzymes, and this is the origin of the sequence. Let's look at the option, then we'll discuss one by one. The first indicate ORI. ORI, that is origin of restriction enzyme. Is that so? No. The ORI clearly indicates that it is the origin of a replication complex, that replication has to start from this site only, right? So this first option is a wrong option. Let's look at the B option, ROP, reduced osmotic pressure. When we are talking about a vector, that means we are looking for one vehicle that help in transferring of a gene. In this, they have written reduced osmotic pressure. This reduced osmotic pressure, here ROP doesn't mean that. ROP means replication proteins. So what are these? Replication protein. Replication protein. Whereas this ACL, BAM, H1, these are what? Restriction enzyme. Restriction enzyme. Restriction enzyme. Now let's look at the option. And similarly, what are these? These ampicillin and tetracycline are the selectable marker. Now, let's come back to the C option. C option states HindIII eco R1 are selectable marker. Is that so? No, they are not at all selectable marker, but they are what? Restriction enzymes, which help in cutting this particular plasmid into the various fragments. And what they will produce? Sticky ends. Normally, what we require for this recombinant DNA formation, the sticky ends. That's why we normally go for those restriction enzyme which produces sticky ends. Now, what are sticky ends? Suppose this is a DNA, double stranded DNA sequence. In this case, the sticky ends are those if at one particular DNA sequence, that means suppose at the 5' to 3' direction, they cut over here. In the second one, suppose they cut over here like this. Now, what I will get is a sequence like this. This is called as a sticky end, or if I elaborate this, I can write this as like this. These are called as a sticky ends, because these sticky ends can easily join to our gene of interest. Similar kind of you can say the treatment, the restriction enzyme treatment will be given to the another form or another DNA as well. In that DNA, the sticky ends will be produced, and we know that for the restriction enzyme, they need a specific site so that it can cut at a specific site. Similarly, in the DNA as well, they will cut at a specific site. The same kind of these I can say that sticky ends will be produced, and that sticky ends will be complementary to each other. So, the chances of binding of our vector with that of the gene of interest will be more, right. So, option C indicate HindIII eco R1 are selectable marker. This is not the right option. Let's look at the option number D, which indicates ampicillin resistance, tetracycline resistance are antibiotic resistant genes. Yes, these are also called as a selectable markers and antibiotic resistance gene, right. So, this was the first question, and for that first question the option D is correct. Now, I have called this as a this is a selectable marker. What do you mean by selectable markers? Selectable markers, suppose what I do, I have these two regions, this regions and this region. With the help of restriction enzyme, what I do, I remove this, right, and I culture these type of cells on ampicillin. Then what will happen? They will be able to survive. When what I do, I treat the cell, the cell which will, recombinant cell which will be formed with tetracycline. Will that cell will be able to survive? No, that cell will not be able to survive, because now, because this tetracycline resistant gene is removed, they will be not able to survive in that, and this is called as a selectable marker. That means cell is not surviving, that means our gene of interest has added to it. So, this was the next question number D. Hope you are clear with the question, question number first, option number D. Now, let's talk about the next question, question number 2. DNA fragments generated by restriction endonuclease in a chemical reaction can be separated by. Very easy question, one of the question which was asked in previous year NEET exam. So, what we have is the DNA fragments which are produced by the restriction endonucleases. Restriction endonucleases are what? These are those enzymes which produce a specific cut. Specific cut they produce. When they produce a specific cut, what exactly they need? They need some palindromic sequences. palindromic sequences. They target that palindromic sequences. They have some specific, they need a specific sequence so that they can easily cut that fragment. So, suppose this is a DNA fragment. In that DNA fragment, various palindromic sequences or the specific sites are there. Later on, what I will get is a number of the sequences like this, like this. Suppose I got 4 sequences. Now, the basic method by which I can separate these sequences, because it's there will be a size difference between these 4. When there is a size difference, what is this? There is a size difference. And we know that the size difference can easily be calculated by using an electrophoresis. In the electrophoresis, we pass those sequences of the DNA in the gel, and we pass electricity. And when we pass electricity, the larger molecule, the sequences which is having a more nucleotides, they will not be able to walk fast, they will remain over there, or they will take a very small distance from the original position, whereas those which are smaller, they will move fast. As they will move fast, what I will see that bands will be obtained. The last band, which is away from the origin, or you can say from that groove in which I have placed our DNA sequences. If it is far one, that means it is a smaller one, whereas those band which is closer to our main hole, I can see that this is a bigger one. So, out of these, electrophoresis is the correct option for the separation of these DNA sequences. The first indicate polymerase chain reaction. Polymerase chain reaction is that chain reaction in which a DNA sequence which we have is a small length, have a specific DNA, specific nucleotide sequence. In this PCR, what I do, I prepare a copy of that particular DNA sequence. So, this is not related to the restriction fragments. Next is the restriction mapping. Restriction mapping could not be done like this. Restriction mapping is just like a molecular marker technique by which we can determine that such restrictions sites they are present, and these sites are specific for one type of restriction enzymes. Centrifugation, we all know the centrifugation, that means what we are doing, we are separating those sequences, not sequences, we are separating the palette as well as supernatant. And this separation can only happen if there is a proper difference among their density, right. If now come at this side, if I got these 4 sequences like this, and these 4 sequences, suppose they have a very small difference among them, 1 or 2 nucleotide difference is there. How can you separate this with the help of centrifugation? Because at the end of centrifugation, what I will get is a palette, and it is very difficult to separate this because it is of very small size. You know that it is also difficult for us to separate DNA, but though we can do that, we can separate the DNA with the help of centrifugation process, but these restriction fragments are like a very small one. Very small nucleotide sequence, and the separation of this with the help of centrifugation, it's a very tedious process. It's next to impossible, so this one is the wrong option. So, the correct option for the question number 2 is a B. Let's focus on the next question, question number 3, for transforming. For transforming microparticles coated with a DNA to be bombarded with the gene gun are made up of. Now, they are discussing about the gene gun. Gene gun is a method of transformation. In this, what we do, gene gun is a method of transformation. In this, what we do, it's like a gun, look like a similar like that of a normal gun. In this, what we do, we take a small beads of metals, and basically the gold and tungsten is taken. Gold and tungsten is taken, and that golds and the tungsten particles, they are coated with the DNA of interest, those sequences of interest. What I do, I bombard these particles, the gold and the tungsten particle, at a very high frequency, at a very high speed to our target. What I have over here, I have a unilayer of cells like this. With the help of gene gun, what I do, I target such type of, you can say the particles at a very high speed. And those cells which are present over here, these they can enter into such type of cells, and basically the plant cells, they are used for this transformation experiment. Normally they are we can do that with the help of these gene gun, and normally we go for plant cell transformation, right. So, what we need, a gold and the tungsten beads we need. Now, come back to the question. A indicates the silver and the platinum or zinc, silicon, platinum, and the answer is the gold and the tungsten is used. So, the option 3 for the question number 3, the option is option D. Now, let us focus on the question number 4. This is also one of the good question. I do not remember the exact year in which it was asked, but this is quite basic also, but a good question. Plasmids are suitable vector for gene cloning. Why? Now, what is plasmid? Plasmid is an extra chromosomal DNA. Extra chromosomal DNA. It is an extra chromosomal DNA. Plasmid, they act as a vector, they act as a vehicle so that they can transfer our DNA of interest. Now, let us look at the option. Option A indicate are small circular DNA molecule. Yes, they are circular DNA molecule which can integrate with the host chromosomal DNA. Is that so? It does not integrate into them, they have their own replication origin. They replicate independently of our chromosomal, of our bacterial chromosome. So, this option, option number first is incorrect. Let us look at the question number, option number B. They are the small circular DNA molecule with their own replication origin site. Yes, this is the right option. Let us look at the option number C, can shuttle between prokaryotes and the eukaryote cells. Is this specific for the prokaryotes? Is this specific for the eukaryotes? No, not at all. So, this is a wrong option. Often carries an antibiotic resistance gene. Yes, they do carry this antibiotic resistance gene, but the correct definition is they are small circular DNA molecules with their own replication origin site. So, the B is the more correct as compared to the D option. Which one of the following markers uses of RNA as a template for the synthesis of DNA? So, the question saying I have to look for that enzyme. Suppose this is an RNA and that RNA is converted into the DNA. RNA is converted into the DNA. When I have to convert this RNA, it's a DNA into the RNA, I know that what I need is a polymerases. That polymerases, they come, they act on this DNA and they convert this into the RNA. For the reverse conversion, for the conversion of this ribonucleic acid into the deoxyribonucleic acid, a reverse enzyme is used and name of enzyme is reverse transcriptase. Reverse transcriptase. Reverse transcriptase they need, so that the RNA can be converted into the DNA, right. Now tell me one thing, in which sequence, in which specific virus this reverse transcriptase is present, that is a HIV virus. We have another examples also, but this is one of the good example, HIV, which is responsible for the AIDS. So, they are saying the question and correct option is option number A for the 5th question. Let us look at the next question. Which one of the following is not a restriction enzyme? Now, we know that the restriction enzymes are those enzymes which have a specific site for cutting the DNA. They need a palindromic sequences. EcoR1, HIND3, PST1, they all are restriction enzymes. restriction enzymes. In the whole session, I have given you one list of restriction enzyme and the sequences for which they are responsible for cutting. I have already given you, among that, this EcoR1, HIND3 and the PST1, they are some specific enzymes. Other enzymes are also there like BAMH1, they also come under this category. Similarly, EcoR2 is also an example. The D option indicates the DNase. The DNase is also an enzyme which cut the DNA, but the difference between the restriction enzyme and the DNase is that they cut at non-specific position. Cut non-specifically. They can cut anywhere, whereas this restriction enzyme cuts specifically at particular site. Specifically at particular site, right? So this is the difference between a normal restriction enzyme and the DNase. So the correct option is option number D. Let's look at the next question, question number 7. This is also related to your restriction enzymes, restriction endonucleases. This question indicates that GAATTC is a recognition site for the restriction endonucleases. In the whole sessions of this chapter, I have already given you one chart. In that chart, I have given of restriction enzyme chart, the endonucleases and exonucleases, that these are the certain different enzymes and they have a specific sequences on which they bind. I have already given those palindromic sequences, and each of these enzymes, they have a specific palindromic sequences. So do one thing, take out the chart again. Hope you remember, hope you have seen those sessions, and in that session you will be able to remember that GAAT is that sequence which is identified by EcoR1, right? This is one of the important question. So by this, why I have taken this question, so that it's your duty to remember all those sequences because they are very, very, very important. Also, you have to remember that which of these enzymes they produce sticky ends, which of these enzymes they produce blunt end. Why we prefer the sticky ends over the blunt ends. So this is the sequence. Now the question number 8 is also related to the restriction enzyme. They have given certain sequences. Let's look at those sequences. Let's read the question first. Which one of the following palindromic base sequences in the DNA can easily be cut at about the middle by some restriction enzyme? What I'll do, I'll take these options one by one. Let's look at the first option. For example, first option, let's do one sequences runs in 5' to 3' direction. The sequences says the C, G, T, T, C, G, and this is a 3' end. Similarly, another we have is a ATC, G, T, A. Now tell me one thing, are these palindromic sequences? Palindromic sequences are those sequences which we read in any of the direction like this or this, they should come out to be same. But these sequences are not coming out to be same. For example, CG, TT, CG. Is that coming out to be same? No, not at all. So that means a sequence is a wrong one. This is not a palindromic sequences. What we need for restriction enzyme is a palindromic sequence. Let's look at the second option. Second option indicate at the 5' end we have G, A, T, A, T, G, and this is a 3' end. Similarly, CTA, 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 right? So this should be 5' over here. So 3' to 5'. Now tell me one thing, these are palindromic sequences or not? No, these are also not palindromic sequences because when I am reading this from this frame or this frame, they are coming out to be different. There has to be G over here and it has to be A over here, and that you can easily correlate because the complementary sequence of G should be C over here, right? So this is some wrong option. Let's look at the third option, the C option, which indicates GAATTC and another CTTAAG. Now this is also 5 prime to 3 prime. This is also 5 prime to 3 prime. Now let's read this GAA. These are palindromic sequences. So the correct option is option number C for the 8. Even let's look at the option number, the fourth option, which indicate we have CACG TA, and below we have CTC AGT. This is also a wrong option because they are making this C complementary to C, which is a wrong one. And here also, so this is a wrong option. So correct option is a C one. Let's talk about one of the palindromic sequences, or common palindromic sequences, that is MDAM, madam. This is a palindromic sequences. Hope you are clear with this palindromic sequences. Why I have chosen the C option? So this was the question which was asked in NEET exam when it was called as an All India PMT exam. This was asked at that time, I guess so. Now, the next question, question number 9. It's related to PCR. First of all, tell me one thing, who discovered PCR? PCR was discovered by Carey Mullis. Discovered by the Carey Mullis, and it is that sequence, it is that procedure by which if I have some specific DNA sequence, and what I can do with the help of the PCR, I can prepare a copy of these sequences. N number of the copies can be produced, and the whole process is called as a PCR, polymerase chain reaction. The whole PCR method is based upon 3 following steps. One is called as a denaturation, second is annealing, and third is synthesis. First is called as a denaturation. In the denaturation process, what we do, if I have a double sequence, a double stranded DNA, what I do, I'll make this double stranded DNA into the single stranded one. Second is called as annealing. Now, what do you mean by annealing? We have primers with us, and primers, for the DNA synthesis we need primers, and those primers we allow to bind those primers over here like this. When these primers they bind like this, the synthesis can start. So annealing means annealing or joining of primers. Joining of primer, and third is called as a synthesis phase. In the synthesis phase, we uses restriction enzyme, because restriction enzyme, they need one end over here, we need an OH group over here, so that they can extend those strands. When they extend those strands, we can produce a copy of it. And with each cycle, initially this was a problem when it was the PCR was first discovered, that they have to add polymerase again and again, because in the first step we have used the temperature higher. And in that higher temperature, this is called as a denaturation, the 2 strands separate. And we know that we are using enzyme over here, and that enzyme is made up of protein. So what we need that particular enzyme that should be resistant to higher temperature, so that it should not denature. In that case, the certain modification is done in this whole process, and they have used one enzyme which is called as a Taq polymerase, and that Taq polymerase was and this Taq polymerase was obtained from Thermus aquaticus. And this particular bacteria, they can live at the high temperature, their enzymes, they are resistant to high temperature. So that's why they have extracted, they live in a hot spring, that's why they have extracted such type of polymerases in this. Now let's come back to the question. Our main question was very simple. This was based upon, we have to look for the correct sequences in the particular order. The correct option is A, denaturation, annealing and the synthesis. Hope you are clear with this. Let's talk about the next question, question number 10. Another good question. We have to look for the option which are correctly matched. Let's start from the last option. Hind 2, are what? They are restriction enzyme, we know that, and it is written over here, they act as a plasmid or the vector. No, they are not plasmid or the vectors. What are these? Restriction enzyme. Restriction enzyme. Ligases are those which help in joining the sequences of the DNA, we know that, but in the another column it is written over here that they are molecular seizers. Molecular seizers are nothing, they are restriction enzymes, whereas ligases are what? Ligases are they basically help in joining the DNA. So this option and this option is the wrong one. Let's look at the option number C, which indicate PBR 322. PBR 322 is nothing, this is just a vector which help in carrying a DNA, and this was extracted from the E. coli. Very efficient vector. So the correct option for this should be like this, there should be plasmid vector, but this is also a wrong one. In the second column in front of this the enzyme is written, but these are not enzymes. The B indicate thermus aquaticus. Thermus aquaticus, I have already discussed that it produces one enzyme which is called as a Taq polymerase, and these Taq polymerase, this is used in the PCR process at the synthesis time. So in the correspondence it is written BT gene, so this is also a wrong one. BT means Bacillus thuringiensis. BT gene is that gene which provide resistance to the plant. So let's talk about the first, Agrobacterium tumefaciens. They produces crown gall disease. Crown gall disease. It produces a tumor-like condition, and they are used for the transformation process because they can easily infect the plant cell and they causes tumor over there. By use of non-pathogenic Agrobacterium tumefaciens, we can use it in a transformation process. They incorporate their own DNA into that plant cells. So when they incorporate the DNA into the plant cells, that DNA get accumulated in the plant cells and they replicate with the help of a host machinery. When they replicate, they produces the tumor or the crown gall disease, and this is used for the transformation process. So the correct option for the 10th question is the option number A, that is Agrobacterium tumefaciens. Right. So students, with this we have completed the 10 question which is related to the chapter Biotechnology Principle and the Processes. Hope these 10 questions are clear to you. Hope you are clear with this, that such type of questions they used to ask, and what you have to learn, how you have to prepare yourself while before appearing for the NEET exam. Students, so this is all about today's session. We'll meet you next session.